Okay, hello everyone. My name is Rihanna Rajkumar and I'm an international attendee at the Biomed Bootcamp. And today I'll be discussing the topic of genetics. So before I begin, the slide shows the four main objectives for this lesson, which includes genes, mitosis, meiosis, and genetic engineering, which will all be linked to different diseases in the medical field. So to begin our lesson today, we have to define what a gene is. So a gene is defined as the functional and physical unit of heredity, and it's passed from a parent to its offspring via chromosomes. And a chromosome is basically a thread-like structure inside the cells of a living organism that carries genetic information. And it's made up of DNA, proteins, and it contains all the instructions, or in other words, genes, that determine how a body grows, develops, and functions. And they're typically found in the nucleus of cells and are arranged in 23 pairs. Now, each chromosome of a homologous pair carries the same set of genes. Therefore, each body cell has two copies of each gene. So it sounds confusing, but when you look at the diagram, it actually makes sense. So it's not too clear right now, but if you zoom in a bit, we can see that there are different sections in the chromosomes that are being showed below. So let's take the top section, for example. That section is in the color blue, and they describe that as the gene for eye color. So if we follow the arrow, because we know that we get our genes from mom and dad, right? So we have two different chromosomes. So if we follow the arrow to which is pointing to those chromosomes, we can see that it's different for those two. Even though at the top it's the gene for eye color, each chromosome has a different color for the different organism. So let's call the first one the male chromosome, and that gene for eye color at the top is blue. However, the second one is brown. So even though it's the same gene for eye color, the eye color can change depending on the organism. It's the same gene, but depending on the organism, the color of that eye would change, and vice versa. Okay, two terms that we need to familiarize ourselves with would be homozygous and heterozygous. So if an allele of a gene is the same, we call it homozygous. However, if the allele of a gene is different, we call it heterozygous. And well, this will make sense when we delve into the topic of dominance. Okay, so do you see the letters in the circle? Those are examples of genotypes. A genotype is basically the genetic makeup within the cells of an organism. So in the previous slide, we met the terms homozygous and heterozygous, which are basically what we call different genotypes. So these genotypes, apart from being homozygous and heterozygous, they can also be dominant and recessive. And this occurs when the allele of an organism is either expressed or if it's hidden. So obviously, if something's dominant, you'll be able to see it more. But if it's hidden, you wouldn't really see it as much, so that's recessive. And based on how visible that is, that's also called the phenotype. That's the observable characteristics of an organism. So usually this is represented by capital and common letters. Capital for the dominant and common for the recessive. So in the presentation, we see three genotypes: homozygous dominant, heterozygous, and homozygous recessive. So homozygous dominant is represented by two capital letters. So for example, in this presentation, we use the letter B. So this is due to the genes being the same. So we use the same letter, and both genes are dominant in their respective parents. That's why we make it capital. So it's the same gene plus it's dominant, hence the reason it's two capital B's. Homozygous recessive, however, is represented by two common letters. So usually common letters are used to represent recessive genes. So even though they're homozygous, which means that the genes are the same, they are recessive. So we represent them by two common B's. And then heterozygous consist of dominant and recessive genes. So both of the alleles are different. So for example, let's say we have a dad with brown hair, and that's dominant, and a mom with red hair, and that's recessive. So when you do a genetic cross or a test cross, you will end up with a heterozygous genotype. Hence the reason it is a capital B and common B. Capital B for the dominant brown hair trait from dad, and common B for the recessive red hair trait from mom. And obviously, because the capital B is the dominant one, that's what the organism will express when it's born. That's going to be the phenotype, the brown hair, not the red hair. So there are different types of dominance as seen in the presentation here, namely incomplete and codominance. Now this is easy to explain, right? So let's think of a color palette. Let's say you're going to paint something, and we have really bright red paint and we have white paint, and we want to make a pink heart. Obviously, you'll take the red and the white and you'll mix them together, resulting in the pink. That's incomplete dominance. That's just where the alleles blend together. So for example, this pink flower could have been made from a red genotype and a white genotype, resulting in this. Whereas codominance is an expression of both. So instead of blending, it's just kind of there. So instead of having a pink flower, we have a red and a white flower, which is also amazing. It's just that the alleles don't blend in this case. Hence the reason we call it codominance, because they coincide with each other. So another thing to note would be that there are also sex-linked characteristics. So we know that females have the XX chromosomes, whereas males have the XY. So usually in sex-linked characteristics is usually by the X chromosomes and not the Y, because a segment of that Y chromosome is actually missing. So the offspring gets these characteristics from the X chromosomes exclusively. So an example of a sex-linked characteristic is the disease hemophilia, which deals with the irregular clotting of blood. So as we can see, there's a small table over here with the respective genotypes and phenotypes. So if you observe closely, there are dominant and recessive genotypes linked to the respective chromosomes. And if we look at the last row, the Y chromosome has nothing attached to it because a segment of the Y is missing, as we discussed before. So one thing I just want to throw in here as we're on the topic of dominance and recessive and dominance and all these different genotypes and phenotypes would be sickle cell anemia, which I'm sure we've all heard of. And this is actually caused by mutations where the red blood cell takes the form of this kind of sickle shape. So I just wanted to show you all the genotypes and the phenotypes of sickle cell anemia. So in the event that you ever have to do a genetic cross diagram, you'll know how to do it. Okay, so now we can move on to mitosis, which was our second objective. And well, mitosis is a fairly simple process to understand. So let's begin. So in the cell cycle, there's a sequence of events that occurs between the start of one cell division. So in other words, mitosis, right? And mitosis is divided into 4 stages: prophase, metaphase, anaphase, and telophase. And some people might know it by the acronym PMAT. So before mitosis, there's interphase, where the cell is prepared for division, and the chromosomes are shorter and fatter, and it makes an exact copy of itself, forming 2 chromatids joined at a centromere. And in prophase, the chromosomes are made visible, so the nucleus shrinks and disappears, and the centrioles move to the opposite sides of the cell, and the spindle fibers form, as we can see over there. In metaphase, the chromosomes line up along the equator of a cell. Anaphase is where the chromatids separate and move to opposite sides of the cell. And in telophase, the chromosomes unravel and the nuclear envelope forms around each group of chromosomes, to make two nuclei, resulting in two identical nuclei being formed. And after all of this occurs, a membrane develops down the middle of the cell to divide it into two, and two identical cells are formed. And that's also known as cytokinesis. So I have the stages of mitosis in point form for those that think I was talking a bit too fast, because I talk fast, so I'm very sorry. So you can always pause the video to look at it whenever you get the time if I was explaining too fast. So sorry about that. So now we're going into meiotic diseases. Okay, so the first meiotic disease we're going to talk about would be mosaic variegated aneuploidy. I know it's a lot as a mouthful. So it's also known as MVA, which is much easier to remember. And it's a rare genetic disorder characterized by the presence of cells with varying numbers of chromosomes in multiple tissues. So this mosaicism leads to a mix of normal and aneuploid cells. And this can cause things like growth retardation, developmental setbacks, and even risk of cancer development because of the unstable amounts of chromosomes in that organism. And this disease is often autosomal recessive, requiring mutations from both parents at the meiotic level. And another meiotic disease that we can look at is Mulibri-nanism. And it's a rare genetic disorder affecting growth and development caused by mutations in a specific gene. And the name, which is actually really cool, it stands for muscle, liver, brain, and eye, reflecting the organs primarily affected, which is very creative. So cool. So persons with this disease can experience growth deficiencies, abnormal organ development, and developmental and hormonal issues. And this disease, like mosaic variegated aneuploidy, is autosomal recessive, and it's caused by that mutation in that specific gene. So now we're moving on to meiosis, which is our third objective today. And it's not really that difficult to understand as well. Once you have an understanding of mitosis, you can understand meiosis. So I'm not going to delve too deep into this because I explained mitosis before. So I'm just going to give a brief overview of it. So at the beginning of meiosis, each chromosome forms two chromatids, and it's joined by a centromere, as in mitosis. And the homologous chromosomes then come together. So there are now four chromatids close together. And genetic information is exchanged randomly between these chromatids, and this is known as crossing over. So in metaphase 1, the homologous chromosomes align randomly across the equator of the cell, and then the members of the homologous pairs separate and move into opposite sides of the cell. And the cell then splits to form two cells, and the division repeats. So that's how we get prophase 2, metaphase 2, anaphase 2, telophase 2. So the division repeats with the chromosomes again lining up randomly along the equator of the cell, only that second time around the chromatids separate, not the chromosomes, resulting in four daughter cells, each with different genetic information. So yeah, that was a lot to digest and mouthful, but don't worry, I have slides explaining everything from meiosis 1 to meiosis 2. So if you guys think I was talking a bit too fast and you didn't understand, you can also always come back and pause the video and look at the different points on meiosis 2 and meiosis 1 to make sure that you understand it. So like mitosis, we're going to talk about some meiotic diseases, namely Klinefelter syndrome, which is where there's an extra X chromosome in a male, leading to a 47XXY karyotype. And this results from non-dysfunctional disjunction during meiosis, where the chromosomes fail to separate properly. So it could result in tall stature with long limbs, as seen in the diagram, well, the picture, sorry, and reduced muscle mass as well as body hair and enlarged breast tissue. And it can also result in delayed speech and language development and learning difficulties, especially in reading and writing. However, unlike those diseases that we saw in mitosis, this one is not inherited. So another meiotic disease that I'm sure a lot of people are familiar with would be Down syndrome, and it's actually a genetic condition caused by the presence of an extra copy of chromosome 21. And this results in a total of 47 chromosomes in an affected individual instead of 46, as we discussed before. And the normal development of this individual is disrupted. This includes their physical, emotional, and mental development, so they might find it a bit harder to learn, their growth is a bit stunted, and they have a variety of physical and cognitive characteristics, as we can see in the picture right there. Okay, so the last thing I want to touch on would be genetic engineering, and it probably doesn't sound as impactful right now, but you'll understand as we go a little bit deeper into it. So, biotechnology is the science which involves the harnessing and exploitation of biological processes, systems, and organisms in manufacturing industries. And an organism that has genes added to it from another species by genetic engineering is known as a transgenic organism. So, diabetes. You're probably thinking, what on earth diabetes has to do with genetic engineering? Well, we'll see in a bit. So, just a bit of background into diabetes. It's a chronic medical condition where the body can't properly regulate sugar levels. And this happens because of either insufficient production of insulin or the body's inability to use insulin effectively. And there are different types of diabetes. So we have type 1 and type 2. And type 1 involves where the body's immune system attacks insulin producing cells in the pancreas. However, type 2 is when the body becomes resistant to insulin or it doesn't produce enough. And there's also a third type called gestational diabetes, and that's usually one that develops during pregnancy, and it can increase the risk of type 2 diabetes. So now, the fun part. This is where genetic engineering comes in. So insulin production, we can actually make insulin from bacteria with the use of genetic engineering. So if you look at the diagram, we have normal bacterium cells with a human cell, and there's plasmid inside of that bacteria. So that's that little circular DNA in the bacteria. So plasmid itself is cut using specific enzymes, and there's DNA being extracted from human pancreatic cells, and they combine these two things together. So they take a piece of the plasmid and they take DNA from that pancreatic cell and they combine them from two different species. And now that plasmid or that recombinant DNA, because that's what we call them when they're combined, recombinant DNA. That plasmid is introduced into a bacterium, and then that bacterium multiplies, and the genes are expressed to make their own different parts and such. And that insulin gene itself, which was inserted, would also be expressed. So the plasmids itself are being mass produced as the bacteria multiplies, and the insulin gene is also being mass produced. And insulin is produced when the gene is expressed, and it's separated now. Like in the final spaces, we extract the insulin and it's separated and purified, and it's used in diabetic patients. So wow, look at that. Isn't that so crazy, so amazing? So that's how genetic engineering affects the medical field, by insulin production. Another application of genetic engineering, it could also be used to help treat some hereditary diseases. I didn't include it in here, but this is just like a fun fact. So it could be used to treat some hereditary diseases in humans, like cystic fibrosis, which is a disease that affects 1 in every 2,500 babies. And it's caused by that recessive allele, which makes the mucus in the lungs thick and sticky. So genetic engineering can help us treat cystic fibrosis. So that's extremely cool. So maybe in the future, one of you watching this video would be able to develop genetic engineering so much that we could find a cure for everything. So that brings me to the end of my presentation. Thank you guys so much for listening. If you made it all the way to the end, kudos to you for listening to me up. I hope you guys enjoyed and learned something, and thank you for watching.